In lecture last week, we went over the phases of mitosis. I would expect you to be able to draw those without any help. So if I tell you draw a cell with a diploid number of 4 and draw all the stages of mitosis for that cell, you should be able to take all that information and draw it, label the kinetochore and non-kinetochore microtubules, and show me what would happen at the end of mitosis. What would the results be? What would happen during each phase of mitosis? What are the major events? What are the chromosomes doing? Where is the nuclear envelope? Things like that. Now, when we reach the end of, let me step back, of telophase, we're going to divide in half. And animal cells will pinch in half. Plant cells will build a new cell wall. They call this a cell plate when it's just forming. And at the end of telophase, the nuclear envelope reforms, nucleoli reappear, chromosomes decondense, kind of back into chromatin again, and the spindle goes away. Then cytokinesis kicks in as well, or while this all is happening, cytokinesis kicks in, which is the division of the cytoplasm. And there are some videos that go along with this. I'm going to play this one on here and hope that it comes through on this video. We'll give it, I don't need to loop it. We'll give it a try here.
In animal cells, cytokinesis begins with the formation of a cleavage furrow. At the site of the furrow, a ring of microfilaments contracts, much like the pulling of drawstrings. The cell is pinched in two, creating two identical daughter cells. In plant cells, cytokinesis begins when vesicles containing cell wall material collect in the middle of the cell. The vesicles fuse, forming a large sac called the cell plate. The cell plate grows outward until its membrane fuses with the plasma membrane, separating the two daughter cells. The cell plate's contents join the parental cell wall. The result is two daughter cells, each bounded by its own continuous plasma membrane and cell wall.
Alright, so hopefully you could hear that well enough. If not, I'm sure you can find it within Mastering or on YouTube, something similar. Either way, what's going to happen now is in animal cells, the microfilaments, which are actin filaments, contract, kind of like the strings on a on a hoodie, on a drawstring hoodie, to form a cleavage furrow, which pinches the cell in half, forming two new daughter cells, which are exact copies of each other. Now, in the case of plant cells, the smooth ER produces vesicles that contain cell wall forming material, and those vesicles fuse together, forming a cell plate, which once it touches both ends of the cell becomes the new cell wall. The rest of the cell wall is made more stretchy during this time so it can expand and grow, and allowing the cells to get bigger as they need to be, because the plant has to grow now to get those cells up to full size. So you can see these actual stages, and you'll do this in lab, but you can see the actual stages of mitosis in a microscope. We will not distinguish between 1 and 2 with actual photographs here, though, prophase and prometaphase. But you can see in prophase, the chromosomes are condensed or condensing. The nucleolus is kind of depolymerizing, going away. By the time we hit prometaphase, the nuclear envelope is gone. The chromosomes are condensed. We can't see what's happening with the cytoskeleton here, but you can refer to the previous slides to see what happens there. In metaphase, the chromosomes are lined up at the metaphase plate. In anaphase, the sister chromatids are separated into daughter chromosomes. And then in telophase, we're forming new nuclei, and we are undergoing cytokinesis. In this case, a cell plate is forming between those two nuclei. Now, do remember, though, that in bacteria, this is a different ball game. We have binary fission taking place rather than mitosis, because their DNA is a ring. It might be folded up a lot to fit in the cell, but it's still a ring. So we're going to attach helicase to the origin of replication. DNA polymerase 3 is going to begin copying that DNA until we form two new rings of DNA, and the cell will also pinch in half, resulting in two new daughter cells. And that's how your average prokaryote is going to divide, and they don't usually do anything resembling sex. So how do they have diversity to maintain? How do they maintain diversity in order for evolution by natural selection to occur? You may ask yourself. And let me turn my volume on my microphone down a bit so I don't overwhelm you here. So what they do is they have enough random mutations during mitosis to increase genetic diversity, and there's also some gene transfer between some DNA transfer between bacteria and things like that, which you can learn about in micro. But they do maintain diversity simply through the process of mutation. I really wish you could have all done this. Is a PowerPoint from a few years ago. I had a bunch of students who participated in the pseudoscience fair, and our pseudoscience fair is happening after Thanksgiving this year. We didn't get to participate this year, but it is going on, and I really encourage everyone to attend and check it out. I'll have more information after Thanksgiving break on that. All right, so let me skip this stuff. All right, so I said no. You're not doing that. You're not doing this. This is for a different class. Okay. Now, for mitosis to work properly, it has to be controlled. The cell cycle has to be controlled, and what we do as mammals, and pretty much all organisms have some kind of cell cycle control like this, but we have these series of checkpoints, we call them. One of the most important ones for us is, as animals, is the G1 checkpoint. So remember, most cells are in the G0 phase, just doing their job. And this diagram, what it does is it's trying to show you like the dial on a washing machine, an old school washing machine, kind of relating that to the cell cycle. So it's this knob that's rotating, and these checkpoints prevent it from rotating past that position unless the checkpoint gate is lowered. So in animal cells, once they've divided by mitosis, they grow and they do their thing. That's G0 phase. But if the cell needs to divide, then the G1 checkpoint will be lowered and we'll enter into S phase and copy our DNA. We'll enter into G2 phase and grow a little bigger and copy our organelles. And then we'll hit the M phase checkpoint, or the G2 checkpoint in this case. And that if the cell is told to divide again, if everything checks out, then that checkpoint will be lowered and we'll enter into mitosis. So checkpoints are, as the book kind of shows you in this figure, kind of like stop signs, go ahead signals. Go ahead and divide or don't divide. If the cell is told to divide, the checkpoint is lowered and we enter into S phase. If the cell is told no, do not divide, it enters into G0 phase and just does its job. Now, oh, my text is kind of slid off here. Let me, there's my little boy, by the way. All right, let me fix that. Okay, so here we have a molecule known as cyclin and cyclin-dependent kinase. Okay, kind of a long word. These two molecules stick together to create a mitotic or mitosis promotion factor, MPF. A lot of words here. I'm not going to get too hung up on the terminology. I want you to see how this works, though. So what we have here is a molecule with a lot of phosphates on it. Phosphates tend to stick to proteins, change their shape, and turn them on or off because they have a lot of energy associated with them or a lot of charge associated with them. So what the body does is it produces cyclin. Cyclin is this molecule that can turn those checkpoints on or off, or basically, lower the checkpoint. But it can't do it without a cyclin-dependent kinase. So both of these things have to be stuck together in order for this thing to have the right shape to interact with the enzymes that allow the cell to reproduce. So cyclin-dependent kinase only works when stuck to a cyclin. If you need a little terminology for this to make sense, a kinase is a molecule that phosphorylates things, that sticks phosphate groups onto things. Okay. All right. So what will happen, you can see this graph here, that when cyclin concentration is spiked, MPF activity increases, so the mitotic promotion factor increases, which means the checkpoints are lowered. Okay. What happens is the cell has to have a way to turn these checkpoints on and off. So what it does is it has this molecule, cyclin-dependent kinase, that doesn't do anything until the cell makes cyclin. When the cell makes cyclin, we form MPF, which then allows us to lower these gates, these checkpoints, and allows the next phase of the cell cycle to turn on. So what happens is cyclin sticks to cyclin-dependent kinase, then we phosphorylate enzymes that then turn on, which then allow the cell to begin mitosis, for example. And so this is one way to regulate the cell cycle. Now, issues arise when we lose control of the cell cycle. So cancer cells often don't need the signals that normal cells do to divide. For example, they don't need them. They've mutated in such a way they don't need them, or they make their own signals and they tell themselves to divide and they just ignore the rest of the body. So cancer is really weird. It's your evil twin, literally. It is your cells that have gone off and are ignoring the rest of your body, and they're dividing like crazy, and they've lost cell cycle control. And they're hard to defeat because just think about it. Like let's say you have a bacterial invasion. Evolutionarily speaking, you and bacteria are very distant on a tree of life, right? Very distant. So there are drugs that will kill bacteria but leave you alone. Now let's move up the tree of life to something more evolutionarily close to you, like a fungus. You have athlete's foot or something. Those we can still target and kill, but the drugs might make you a little more sick because fungus is a lot more close to human than bacteria is. Okay? Move up into like an intestinal parasite, a worm in your stomach. All right? That worm is a lot like you, biologically speaking, and it's going to take a drug that could very well make you very sick to kill that worm. Maybe not in every case. Maybe we found a pathway that can target something the worm does that we don't do, that we can exploit. Now, when you get to cancer, you have to kill cells that are you. They are literally your evil twin, and they're dividing like crazy. So it's a challenge to find a way to defeat cancer for that reason. Now, what happens is we lose control of the cell cycle. So how? Well, one way is maybe the cell constantly makes MPF. Notice in this cell cycle, someone's calling me. Please hold. And we're back. Okay, so I want to show you this diagram again. Cyclin-dependent kinase can't work without cyclin. The cell receives a signal, is told to maybe the cell's growing or maybe the body needs to heal, so the cell receives a signal that says, hey, make some cyclin. It sticks to cyclin-dependent kinase. That turns on the checkpoint or turns off the checkpoint. The cell starts to divide, and then cyclin is degraded and broken apart, shutting off the cyclin-dependent kinase again. Excuse me. A cancer cell might not degrade cyclin, or maybe a cancer cell doesn't need cyclin, or maybe it constantly produces cyclin. There are lots of kinds of cancers, and I am not going to, say there's a one-size-fits-all reason for cancer, but a lot of times it's due to some kind of loss of cell cycle control. So remember that Henrietta Lacks's cells have been dividing, her cancer cells have been dividing since 1951. This slide says as long as food is present, some of these cells can just keep dividing. Normal animal cells, let me fix this slide again. Normal animal cells can only divide usually like 20 to 50 times before the ends of the chromosomes, the telomeres are lost and you lose genetic info and they quit dividing. But cancer cells not only have lost cell cycle control, but some cancers also turn on telomerase, and so they become essentially immortal lines of cells, and they just keep dividing and dividing and dividing and invading healthy tissue. And when that happens, then they start depriving healthy tissue of food and oxygen and blood, and they're crowding out the healthy tissue, leading to all kinds of problems. So when it comes to cancer, one cell produced by mitosis or maybe some kind of mutation causes it to be weird, right? And it starts to divide. and this tumor forms, which is basically a series of clones of that cell that's gone wrong. And this tumor starts invading healthy tissue. And then what can happen that's really bad is metastasis, if cancer metastasizes. So that means parts of it start to break off and enter into the lymph vessels or the blood vessels, and they spread throughout the body, and they're like little colonizers. They start new tumors all over the body. So one cell breaks off and gets lodged in a lymph node, and it starts to divide. And now there's a new tumor there. And maybe another piece goes to the liver or the brain, and a new tumor forms there. So cancer that metastasizes breaks apart and spreads throughout the body. So I have a question I ask in lecture. What changes in a cancer cell might increase the chances of metastasizing? Well, think about it. A normal human cell doesn't break apart and float around, right? Like tissue doesn't just come apart. So what might go wrong that could cause that? Well, one thing that might go wrong is maybe there's a mutation that has messed up the extracellular matrix. Extracellular matrices act like glue that hold tissues together, and there's also proteins that hold cells together. There must be something wrong with those structures that allow the cancer tumor to break apart into little pieces. So if you're studying cancer, you might have to come up with drugs that target kinases like MPF. So cyclin-dependent kinase or cyclin, or the MPF factor that's produced when they come together that allows the cell to divide. You might have to target tumors and try to keep them from coming apart. You might try to stop mitosis. You might try to pop membranes of cancer cells. You might do all kinds of things to try to target that cancer. But the trick is finding things that will affect the cancer but won't hurt the patient too badly at the same time. So you should be able to look at this figure from your book and start finding different phases of mitosis. I'll get you started. This cell right here is an interphase. There's the nucleoli. The chromosomes are not condensed, so that's interphase. Okay? Let's see here. Here is anaphase. There's an example of anaphase. Here's an example of metaphase. So start working your way through those. Oh, here's a prophase example, when chromosomes start condensing. So work your way through and just start identifying these as well as you can, because we will do this in lab, and you will have some images on your exam that ask you to identify the phases of mitosis. So here's a question for you. At what point of the cell cycle would you see a chromosome that looks like this? Give your reasons. I'll let you work on that on your own. Meanwhile, I encourage you to answer the questions in the back of the book, work on the guided reading questions for this chapter, draw the phases of mitosis, make sure you understand what diploid means, as well as the various terms we've introduced, and then begin looking into meiosis, because that's where we're going next, and that's where students sometimes get lost. So start thinking about what diploid, what haploid means, what the parts of the chromosomes are. You need all of that information to better understand what's happening when we enter into meiosis. All right, see you soon.